This next section is titled Microbes and Morgellons. I would like to discuss a few thousand patients who have become infected with what appears to be an insect-borne illness that presents with Lyme-like symptoms. These patients also have eruptions of peculiar ulcerating skin lesions which seem to produce fibrous, thread-like substances in varying colors. This mysterious malady has been named Morgellons disease. The additional manifestation of strange thread-like structures complicates disease diagnosis and treatment, but it also seems to support my hypothesis that the engineered microbes of biological controls, or bioremediative tools, may be involved in the epidemic known as Lyme disease and also for Morgellons. Just as Lyme patients are told their illness is all in their head, Morgellons patients are frequently called delusional. In fact, there is a psychiatric diagnosis that is being pushed upon them as delusional parasitosis. This branding of patients with a delusional disorder occurs despite the fact that extremely disfiguring skin and mucous membrane lesions appear on their skin, and the patients display systemic symptoms that are otherwise unexplainable and impossible to manifest upon sheer will alone. The grotesque skin lesions of Morgellons patients are not figments of their imagination, but visually distinctive, ulcerating lesions that produce hair-thin, thread-like proteinaceous fibers. When cultured, these fibers have revealed the presence of the bacterium Agrobacterium tumefaciens, Rhizobium radiobacter. This is the very same bacterium that causes crown gall disease, a plant tumor-producing disease in such plants as ornamentals, fruit trees, and tomatoes. These galls are tumor-like growths that occur between the roots and the shoots of plants and sometimes appear on the aerial branches. This bacterium is also useful as a bioremediation tool and for crop protection against pest insects. A. tumefaciens is often deployed in fertilizer compositions as a biological control. Another pathogen that contributes to the formation of galls is the fungus Endothea parasitica, later renamed Cryphonectria parasitica. This fungi causes chestnut blight, and when it is infected by a tiny virus, galls or tumorous growths are created on tree trunks or branches. Morgellons is currently reported to be infecting thousands of individuals who are unrelated by family and who are located in pocketed areas of the world, not just in one geographical location. The majority seem to be located in the U.S. along the Pacific Northwest coastline and on the Atlantic southeastern coasts where the majority of citrus groves exist, and where the bacteria Agrobacterium tumefaciens is used to address pathogens that threaten these crops. Pathogens that attack citrus are many and include various actinomycetes species of mycoplasma-like organisms such as the citrus Spiroplasma citri of citrus stubborn disease. Another pathogen prevalent in the U.S., in particular on the West Coast, is the fungus Coccidioides immitis. I have covered this fungus and the spiroplasmas earlier and might restate that the skin lesions that accompany coccidioidosis are tuberculin-like and appear to resemble those of Morgellons patients. If we study the geographic regions of patients who present with Morgellons disease, we might determine that these victims live close to areas with high concentrations of biological controls that are deployed against plant pathogens, including the spiroplasmas. We might also realize in some cases the presence of A. tumefaciens in the patient's geographical region may be due to its application in bioremediation, such as on military bases or mines where applied microbes may seep into waterways. Naturally present fungi such as C. immitis contributes to the mixture of fungi present in resident insects. Spiroplasmas and A. tumefaciens may also be naturally prevalent because they are agricultural problems for growers of nuts, beets, grapes, rhubarb, and horseradish plants. I have already told readers that brittle root disease in horseradish plants is caused by Spiroplasma citri. What if an aggregate BB organism contains this spiroplasma strain and one or more fungi to cause the Lyme-like disease called Morgellons? We may question whether the presence of A. tumefaciens is enough to cause the fibrous-like growths which appear as eruptions from the skin and mucous membranes of Morgellons patients. Agrobacterium is a bacterium that is well known for its ability to transfer its DNA into the plants that it infects. Since fungi are unstable and likely to recombine with compatible organisms, it may be that C. immitis and/or A. tumefaciens contribute to Morgellons infections to cause skin lesions. This horizontal gene transferability has made A. tumefaciens an ideal tool for genetic engineering of plants and a useful biological control. Common landscape plants, bananas, and inbred lines of maize or corn have even been genetically transformed using a combination of agrobacterium, sometimes with the addition of Bt cry toxin genes. If we think about it, wool sorter's disease, or cutaneous anthrax, is a mutation of a soil bacillus that also causes skin ulcers, although these look a bit different than the lesions of Morgellons patients, inasmuch as they don't appear to produce fibers. Another factor that some have considered as a contributing factor to Morgellons lesions is the disease tularemia or rabbit fever, which also causes ulcerative skin lesions. The fungi present in the environment, whether through natural transmissions or as applied microbial controls, may be the main contributing factor to Morgellons. Scientists have extensively engineered transfer DNA vectors that have been modified using Agrobacterium tumefaciens in bioengineered plants to study gene expression. Since we know that Agrobacterium can redirect plant genes, it is highly plausible that once insects infected with this organism introduce it into the human body, that the bacterium could misdirect the expression of human genes. If not, the spiroplasmas that infect plants and insects may have their growth redirected by A. tumefaciens before being vectored into a human host. Let's also consider the virus of the gypsy moth Amsactomori, AMEPV. This is an insect entomopox virus that was described by a scientist named Vago in 1963. According to research, this pox virus creates bundles of filamentous structures which are comprised of a protein called P10. These filaments are also called cytoplasmic fibrils. This virus is commonly found in woolly bear, tiger moths, milkweed tussock moths, fall webworms, and other members of the Arctidae family of moths and in their larvae. Many of these insects have been specifically targeted by Agrobacterium tumefaciens containing biological controls. Another class of organism that produces such fibrils are the spiroplasmas. As I said elsewhere, when spiroplasmas are presented with detergents or surfactants such as Triton X100, which is ubiquitous in the world since it is widely used in cleaning products and spermicides, these organisms detach their fibrils and other cytoplasmic contents, which are then free to aggregate with other pathogens, including the fungi. The Autographa californica NPV virus, another baculovirus that has been used extensively in genetic and agricultural engineering, largely to control pest insects, also reportedly creates this same kind of P10 fiber-producing protein. As an example, scientists have genetically engineered a recombinant Autographa californica virus, which they call ACMNPV. From a recent patent, we learned clues to what kinds of structures these viruses can create. Quote, exemplary baculovirus envelope proteins include, but are not limited to, the GP64 protein and its functional equivalents thereof. In one example, the non-paramyxoviral envelope protein includes a full-length baculovirus GP64 protein, such as a full-length GP64 protein of ACMNPV. In addition, the membrane fusion capacity of GP sub64F, which is a baculovirus protein, was tested. GP64, when expressed alone in insect cells, can induce syncytium formation after brief exposure to low pH, such as at pH 4. End quote. This means that these proteins can cause fibrous structures to form. According to a source, syncytia are large cell-like structures that contain many cell nuclei and are filled with cytoplasm. Syncytium is the normal cellular structure for fungi and some algae, including those which form thread-like cells that are partitioned into segments, called dikaryon. The dikaryon is a primitive form that belongs to some fungi, including members of the families Ascomycota and Basidiomycota. The Ascomycota are members of sac-like fungi. Among the members of this subkingdom are brewer's yeast, which is Saccharomyces cerevisiae, the very yeast used in the fermentation process when creating the biological control agent Bacillus thuringiensis, or BT. The P10 protein of the AMEPV, Amsactomori pox virus, is a very late gene promoter that is involved in the creation of virus occlusion bodies. The P10 proteins are known to be important during baculovirus infections and appear also to be involved in the formation of such thread-like fiber or fibrillar structures. Perhaps an insect feeds upon a caterpillar larva that is infected with either ACNPV, AMEPV, or another NPV with this important protein. In the presence of Agrobacterium tumefaciens in the insect's body, pathogens undergo late transcriptional events. This can occur inside the insect or human host, although I prefer to think of the process in the latter. Perhaps this is how the thread-like fibers of Morgellons disease are created, through late transcriptional events that don't occur until inside the human host. This group of pathogens, of course, would be part of the cascade of co-infections that the Lyme patient contracts from an insect vector. Here may be possible evidence that bioagents play a huge role in the determination of diseases which manifest, depending upon the menu of pathogens in the vector once it meets a human amplifying host. For those more technically minded, these P10 structures appear to attach to protein chains during or after transcription at the location of the C-terminus or carboxyl terminus end of the amino acid chain. The C-terminus of proteins can be modified post-translationally, which means after protein translation has already occurred. So after the initial genetic translation or mutation occurs, a secondary process can occur which continues to modify the final genetic form of the organism. Scientists use early and late promoters during genetic engineering to cause certain genetic functions to be performed or suppressed. They can also terminate chemical reactions to prevent late transcription events from taking place. Perhaps a transcriptional process that is meant to take place within plants or within the bodies of the insect targets doesn't get a chance to complete its task before being introduced into the human host. Once there, it completes its assignment only now to the detriment of the human host, who, like the insect, becomes infected with what may now be defined as Morgellons disease. Please be certain to watch Part 2 in this video series.